Hey, today we're going to talk about cell cycle and regulation. Okay, so you are made of cells. Every part of you is cells, and so not all cells are the same age. They last different amounts. So stomach cells, skin cells, bone cells, and some brain cells never die. But this is important because cells do not necessarily live forever for your whole life. So we need to have a way to replicate our cells. Okay, so we need new cells. Our stem cells and some specialized cells make more cells by cell division, so when our cell divides. So why do we need more cells in the first place? So cells get damaged, they get too old to function, and our bodies grow, and we need more cells because of that as well. Unicellular organisms also use this process to reproduce because they only have one cell. Okay, so this is a diagram showing the cell cycle or cell division. So we have different parts that we're going to go through today. So we have interphase, which has the phases of, here this is shown in blue, G1 or growth 1, S, DNA synthesis, and then G2. And then we have mitosis, which is separate from interphase. And that is made of prophase, metaphase, anaphase, telophase, and then we have cytokinesis happening. And all of this makes up the cell cycle. Most of the time, the cell spends its time in interphase because that's why it's shown here as taking up the most time. Okay, all of this cell cycle is signaled like a clock. And the enzymes that do this are called cyclins and cyclin-dependent kinases. Okay, so cyclin is shown here in green, and then cyclin-dependent kinases are kinases, which is an enzyme that phosphorylates. So it's an enzyme, but they're only active if they have cyclin bound to them. So because of this, cyclin can make it so that these CDKs, cyclin-dependent kinases, are active at different times, and that's how we're able to time all of these different processes. Okay, so that's the main thing you need to know, is that a kinase in general is just a term that talks about an enzyme where it is phosphorylating groups, and that is going to activate and deactivate different parts of the cycle. This is kind of showing we have different cyclins then that are going to be active at different parts in the cell cycle. You don't need to memorize which one is dependent where, just the idea that we have different cyclins active at different times because of those cyclin-dependent kinases. So cyclin D can activate certain cyclin-dependent kinases, and then later we can have cyclin E, and those are going to phosphorylate enzymes in order for this timing to happen correctly. An important thing too is enzymes are not destroyed, so they're just activated and deactivated and can be reused. So this is we don't have to memorize it. Okay, so this is showing another way to visualize that what was shown in this graph with the cyclins. Here we have mitosis, G1, S, G2, mitosis, G1, S, G2, mitosis. So this is interphase and metaphase. And here we have different cyclins active at different times, or we have a cyclin active at different times. Okay. Here is also showing that in a circle form, and we have our cyclin-dependent kinase and cyclin forming this complex and being active at different parts. Okay, so that's how all this is regulated. Now I'll get into what happens at each stage. So interphase, this is where the cell performs their regular functions. It replicates its DNA, and it's going to prepare for mitosis. So 90% of the time, the cell spends in interphase just doing its specialized job. Okay, it's like the prep stage, though, where it's living and then going to start preparing. So it's broken down into 3 stages. We have G1, also referred to as gap or growth 1. Here the cell is just doing its function and growing. Okay, the DNA at this point is called chromatin. So it's all spread out, relaxed, uncoiled, because we need to make proteins. We're utilizing this DNA. So we need to be able to access the DNA, and that's why it's uncoiled. Okay, so we also are going to reach a checkpoint, and there's various checkpoints throughout all of the cell cycle in order for us to see if we're going to progress on to the next phase or not. So this is where we're going to decide if it is going to go on or not. So G1 checkpoint is going to check do we even need a new cell? Is this cell healthy enough to divide? Are there enough resources to divide? And then if that is the case, then it's going to go on to the next one. And we'll model this with paper plates in our class. Some reasons a cell might not divide is if there's DNA mutations or damage. No other cells have died, and we don't want, this is showing like a big crowd of people, but you can think of cells the same way. We don't want it to be overcrowded and have too many cells if there's no need for cells, okay? Or if it does more harm than good, like nerve cells, which is why we don't have nerve cells replicating very often, or if it's sick, okay? So if it doesn't pass this checkpoint, the cell is going to just hang out in this G0 phase where the cell does not divide. It doesn't necessarily die, it's just going to continue to function. And at some point, it might get cell signals to put it back into G1 and have the checkpoint again and go on. But it's just going to continue to function until it eventually dies or it gets signal to go back. Okay, where it can re-enter. But it has to have specific environmental cues. Okay, so if the cell does pass the checkpoint, it goes on to the next phase, and that is the phase to prepare for replication. So we're making copies of our organelles, the cell is going to grow, and it's going to produce proteins needed for cell replication and cell division. And it moves into the second part of interphase, which is phase S, or synthesis. This is the replication phase. This is where the DNA replicates, because we're eventually going to need 2 sets of DNA because we're going to have 2 cells. So we need, every cell is going to have an exact copy of the DNA in all of the different cells. Okay, so when DNA replicates, the original DNA is going to be connected to its copy, and that is called, where we connect them is called a centromere. Okay, and so the reason we need to have these copies, the 2 copies connected to each other, is so that we can make sure that when we have the cells divide, we have 1 copy goes in one cell and 1 goes in the other. We don't want 2 copies of the same DNA going in one cell and a different chromosome going in another cell. We want to make sure 1 copy from each goes into each of the cells. Okay, so yeah, that's what we're making. The proteins themselves that make up the centromere are called cohesions, and they hold the 2 copies together. The 2 copies of the same chromosome are called sister chromatids, and they're held together at that centromere. Okay, so here's showing a chromosome, and then we replicate the DNA. We have, this is called the chromosome. So we have 2 chromosomes, and we still have 2 chromosomes, but after replication, each chromosome is made of 2 sister chromatids that are attached to the centromere. Okay, at this point, the DNA does not look like this. It's still just like, looks like a big jumbled string, but this is a helpful way to think about what's happening. Okay. So here again, sister chromatids in the centromere. Okay, after S phase, we're going to go into G2, or gap or growth 2. It's the last phase before cell division. The cell continues to grow, and we're also going to have another checkpoint here. So this G2 checkpoint is going to check to make sure the DNA was replicated correctly in our last step. We want to make sure there aren't any mistakes. If the DNA was wrong, it will try to fix any corrections with enzymes. But if it can't fix any mistakes, the cell will not continue and will go to apoptosis, where the cell will have programmed cell death. So why do we want to kill the cell if it does not have correct DNA? It's because we've already copied the DNA. There's no going back at this point for it to just function normally. It's not going to work. So we're going to just, for the sake of the whole organism, the cell is going to be recycled. Okay, and that's because the error in the DNA. We don't want that error to continue. Okay, if it passes the G2 checkpoint, it's going to move into mitosis. Okay, and we'll do a foldable in class 2 to help us organize all the steps of mitosis. Okay, so the point of mitosis is to make an exact copy of the cell. So we have 1 mother cell, and at the end of mitosis, we will have 2 daughter cells that have exact copies that are passing on that genetic information. So there are 4 phases of mitosis. Prophase is the first phase. I think of P for preparing. It's preparing for mitosis to happen. So the nuclear envelope, the nucleus itself, the membrane around it, begins to disintegrate or break apart because we need that DNA to be accessible. The DNA starts to condense, turning that squiggly chromatin into chromosomes, which we see more here. And to do that, it wraps itself around proteins that are called histones. Okay, so if it didn't coil up, it would be really hard to make sure that we have the DNA going in the correct places because the DNA is all spread out. We want to package in little nice bundles to make it easy to move from one to the other. Okay, so the chromosome here is still made of two sister chromatids. They're just now condensed. Okay, so there's condensed made of two sister chromatids. Okay, the centriole will be created, and organized spindle fibers, which are made of microtubules, will be used to set later with the sister chromatids. Okay, so here's this centrosome that's going to be produced here as well. So the centriole and the centrofibers together, or spindle fibers together, make the centromere. So these are special organelles that are used in mitosis, and they're going to be synthesized in this step. And it begins to move to the poles, so the sides of the two ends of the cell. Okay, the next phase is metaphase, M for middle. So the sister chromatids line up on an imaginary metaphase plate, which is this line in the middle. Okay, by this time, the centrosomes have reached the poles, and the spindle fibers here have attached to each chromosome set at their centromere at the middle. Because the spindle fibers are like the puppet strings that are going to pull these chromosomes apart. The protein where the spindle fiber is attached to the centromere is called a kinetochore. And the spindle fibers help position those chromosomes in the middle. And we have another checkpoint here. This is called a spindle checkpoint. The spindle checkpoint is going to tug back and forth on our chromosomes to make sure that they are lined up properly. Okay, you can think about it as like tug of war here. We're pulling back and forth to make sure that they're all lined up. The next phase is anaphase, A for action. Okay, so once the cell has passed the spindle checkpoint, everything's lined up properly, the spindle fibers will begin to pull and shorten. As they shorten, they're pulling on the chromosomes to pull those sister chromatids apart. The cohesion, remember those proteins that are on the centromere, are broken down by an enzyme, making it so those sister chromatids can be pulled away from each other. The spindle fibers are broken down and shortened, and the cell also elongates. See how it becomes more oval. Okay, the moment the sister chromatids are separated, they're now their own chromosome. So technically speaking, we now have twice the number of chromosomes we had before. Okay, a chromosome is condensed visible DNA, and it's still condensed here because we have them in packages. The next phase, we have telophase, T for termination. This is the final stage of mitosis. This is where the chromosomes reach the poles, the opposite ends, and the nuclear membrane starts to form again around the nucleus, around those chromosomes to form the nucleus. Okay, the chromosomes themselves start to uncoil and relax, turning back into that chromatin form. And before telophase finishes, the nucleus is closed up and the cell enters cytokinesis, where the cytoplasm is going to be split. Okay, cytokinesis, it is not technically part of mitosis, but it is part of the cell cycle. This is where the cytoplasm and the cell membrane are split into two newly formed nuclei, and it happens during the last part of telophase. Animal cells and plant cells have slight differences because plants are going to also need to form a cell wall in between as well. So we call this this part where the cell is starting to break or pinch off the cleavage furrow in animal cells. And in plant cells, we have this plate that's being formed, the cell plate that will turn into this new cell wall. Okay, so the animal cells pinch their cell membrane. Plant cells are going to build that new cell wall using that cell plate. Okay, now we have two identical copies of that same cell. So this is kind of showing an overview of what's happening. These pictures at the bottom are really cool because they are stained pictures. So it's actual microscope slides showing the cell, and the different parts of the cell have been stained these fluorescent colors. So you can see like the spindle fibers in green and the chromosomes in blue being separated. This is showing what the cell looks like. Interphase, the DNA is all squiggly. In mitosis, it's early mitosis. In prophase, the chromosomes are starting to condense. Late prophase, they're all condensed. Metaphase, they're on the middle. And then we have anaphase and telophase, where they're splitting apart and then they're at the poles. Okay, so that's the cell cycle and how it works. If we have issues in the cell cycle where there is a loss of control, this is where we have cancer being formed. So cancer, the idea of the tumors are we have uncontrolled cell replication. So we have mitosis happening too quickly, and when that happens, it forms a tumor. Okay, so that means the checkpoints are skipped, or if we have genetic mutations and things are not flagged properly for apoptosis, that's when cancer happens. Okay, most of the time we're talking about eukaryotes. Prokaryotes have a similar process, but it's much simpler because they don't have as much going on. They don't have a nuclear membrane to dissolve. There's only one strand of DNA. They don't have to worry about having everything lined up properly to separate as well, and they don't have organelles to replicate as well. This process for asexual reproduction purposes is pretty much the same as mitosis, but it has the word binary fission, and it's what we use to describe a single cell organism that's reproducing. It occurs in two phases, the S phase, where the DNA is synthesized, and then we will have the G1 and cytokinesis. So it'll create an identical copy of the parent. And prokaryotes can do this very quickly because it's so simple, and that's also why bacteria can replicate so fast. Okay, so the evolution of mitosis, because prokaryotes evolved before eukaryotes, mitosis probably evolved from binary fission, is the idea. So certain protists exhibit types of cell division that seem intermediate between binary fission and mitosis, is the idea. Okay, that is it for mitosis and the cell cycle. Hope this is helpful. Bye, guys.